So there are a number of really exciting hot areas in biointerface area and a number of recent really exciting breakthroughs. Some of these include in the area of mechanotransduction, so the study of how cells receive mechanical cues from their surroundings. So there's been great interest in understanding how the intrinsic mechanical properties of materials, for example, the extracellular matrix that surrounds cells within tissues, impacts their behavior, and also then how externally applied forces will impact cells and cell behavior. And this has led to some really exciting work recently where it's been demonstrated, for example, that external forces can promote the regeneration of skeletal muscle tissue and bone tissue. There's also been really exciting advances in the area of what I would call immunoengineering, which is the concept that we can engineer the immune system in a human to achieve a desired outcome. And we've seen some really exciting talks at this conference in relation to this. Work, for example, that demonstrates one can induce tolerance in the context of autoimmune diseases like diabetes, that one can promote very effective immune responses against cancer to eliminate tumors and potentially provide a therapy or a cure for patients suffering from cancer. And the development, I would say, of bioactive and dynamic materials, where the materials change their properties in response to cues they receive from their environment. And this then mimics the kind of the dynamic nature of tissues and living organisms and may give us new tools to really control biology.
So in the field of biointerfaces, we expect medical progress and breakthroughs, in particular in fields where we combine biology with technological solutions. Sometimes this is called bionic solutions, where you have, for example, intelligent technical systems combined to living tissues. This is certainly the dream of many of the activities. More closer in time are implants made of stem cells, for example, or materials which have more intelligent feedback loops, such as in artificial heart systems.
So there's tremendous interest today in developing alternatives to the classic animal testing from a couple of different perspectives. One of these is the desire to minimize the use of animals in research, and another is to have models that may more accurately reflect human biology than, let's say, a mouse or a rabbit may. There's a wide variety of different technologies that are being developed in an effort to to have these better models. These include things like organs on a chip that you may have heard of, 3D cell culture models, among others. All of these show great promise and have demonstrated that one can improve our understanding of biology as compared to kind of classic two-dimensional cell culture or mouse models. They are all still a ways away from actually being able to routinely help us move forward in terms of development of medical technologies or therapies. A key issue here is validation. So what still remains to be done for virtually all of these approaches is they need to be validated and demonstrated that the data that they give is truly representative of human biology and whole body biology, for example, by having a thorough characterization and comparison to animal studies. So very promising, a lot of exciting advances, but we still have a ways to go before they become routinely used either in the development pathway for therapeutics or as therapeutics themselves.